Tēnā koutou, kia ora koutou katoa, and welcome, distinguished guests, academic colleagues, students, supporters and friends, to the inaugural professorial lecture of Joe Zuccarello, Professor of Biology in the School of Biological Sciences. My name is Professor Grant Guilford. I'm Vice-Chancellor of Te Herenga Waka, Victoria University of Wellington, New Zealand's globally ranked capital city university and New Zealand's top-ranked university for the intensity of high-quality research. And it's my pleasure to host you all this evening. I'd like to briefly introduce Professor Zuccarello before handing over to him to deliver his lecture. Joe is a renowned phycologist who has contributed extensively to the understanding of the evolution and the taxonomy of algae. He received his BA in Biology and Master of Science in Marine Science from the University of California, Santa Cruz, before going on to complete his PhD on red algal parasites at the University of California, Berkeley. Joe has conducted research on algae in places as diverse as the Netherlands, Plymouth and Austin, Texas. He began his teaching career at the University of New South Wales and moved here to Wellington to take up a role as senior lecturer in Te Herenga Waka in 2004. He became an associate professor in 2012 and a professor in 2020. Professor Zuccarello is extensively involved in advancing the promotion and understanding of phycology worldwide. He has been the president of the International Phycological Society, the president of the Australasian Society for Phycology and Aquatic Botany, and the international vice president of the Phycological Society for America. He has been on the editorial board of a number of journals, including the Journal of Phycology, the Journal of Fisheries and Environment, and Phycological Research. He has made a very significant contribution to his field through peer-reviewed articles, more than 170 at last count, to a range of very high-quality journals, and has written a number of book chapters. He has also presented his research at a number of prestigious international symposiums and conferences. Here at the university, Joe has contributed extensively to the School of Biological Sciences and the university more widely. He became the group manager of the Ecology and Biodiversity Group in 2019, a key role that involves organising and the teaching within the Ecology and Biodiversity program. He is also a committed and passionate lecturer and PhD supervisor. In this lecture, Joe will explore the importance of seaweed in our natural world. He will present his research on algal diversity, genetic variation, reproductive biology and evolution. He will highlight how basic biology is essential for understanding our natural world and its evolution, and why universities are perfect places to pursue these goals. Please join me in welcoming Professor Zuccarello to the podium.
Thank you all for coming. It's nice to, Grant, Ehsan, Wendy, thank you for coming. It's nice to be here and give this talk on some of the work I've done over many years, too many years. Actually, today I was talking to a student and I told them about a paper I wrote, and they said they were 4 years old when I wrote that paper. And so it's kind of scary. So I'm going to talk about investigations of seaweeds, and I'm going to cover a few things. One is sort of the things I've done, sort of the general things I've done, but I also want to highlight how being at a university and having the opportunity, we call it academic freedom, we call it the ability to just follow what we like, is useful and has led to productive insights, at least productive for me. And then I want to finish about on a topic that's a bit more esoteric and that is part of ongoing research. So this is the first slide. And I guess I'll start by how you get around to studying seaweeds. And it's this, right? It's what are these things, right? That was my question. What are these things in the ocean, right? I didn't do it here. I did it in California, but I was just very intrigued about stuff that I didn't know anything about. And so what are these things? Can we name them? These are all relatively large things that people probably couldn't name. Just how can you go through life without knowing the things around you? And so I was just kind of interested in natural history. Sorry about that. Whenever I take these videos on my phone, my son always says it looks like I'm trying to film Bigfoot. obviously I'm not doing it the proper way. Oopsie. So I'm going to talk about my biological interests, and of course it has to go back to the people who inspired me and taught me. And there are a few, and I'll talk about their work. And they've been teachers, and I'm not sure I fit into that mold, but when you think back at the people that influenced you, it is those mentors. And so my approaches are also based on what my mentors taught me. And so then I will also talk about some of the things I study that I find interesting, and then pick up new research directions because I have this ability to do what I like. I hope Grant doesn't mind that he has people around that just do what they like. So one of the first mentors was a woman called Linda Goff. She was at the University of California, Santa Cruz, and her approach to science was very question-based. Guessing, we call them hypotheses in science, but making guesses about things and then trying to think about the organisms you could use to address those questions. They're always seaweeds. The organisms you could use to address those questions. Right? So it was always questions, and so it was very exciting to come up with these guesses and then try and figure out if they were right or wrong, which is great. The problem with having hypotheses that you are wedded to is that you also have to be able to drop them, and sometimes that can be difficult. This is Linda when she's retired. She's very much involved in carving birds, which is a nice thing. We all have to think about what we're going to do when we retire. My PhD supervisor was a guy called John West, nothing to do with the tuna company. He was very different from Linda Goff. He didn't ask any questions. His approach was just look at them, just study them. Don't come up with guesses about how things are done. Just try and understand what they do, see how they reproduce, see how they grow, find out where they grow, and come up with those questions later. Very different approach. This is me with John West as a PhD student. The woman on the other side is Valerie Vreeland. She used to work on, well, she doesn't anymore because they're both retired. She used to work on algal cell walls. That's John, and that's me when I was younger as a PhD student. And of course, with John, we've traveled the world collecting algae. Here we are somewhere in the tropics, a little bit later in life, and here we are in some beautiful tropical paradise, working real hard on university money. This is actually El Salvador. We were on the Pacific coast of El Salvador. It was very nice, with our friend Francisco Pedroche. Students have seen this all the time. This is my approach to understanding the biological world. This is where diversity is, in my view, of organisms, and it has nothing to do with animals. Somewhere on that tree, there are animals. In fact, I can point them out to people. This is where animals are. But the diversity is a whole bunch of other things. These are eukaryotes. We're not even talking about bacteria, which would fill the screen and three more. The algae are all over the place. And basically, they're photosynthetic organisms that have particular characteristics. I'm going to talk about a few of these. I mostly study red algae, which is this group called the Archaeplasidae. Then there's green algae and the large brown algae in the Stromenopiles. But there's algal groups and other things. Touched about them in recent research every once in a while. So there's three main algal groups, the browns, the greens, and the reds. I just came out from an evolution lecture in 329, so I'm going to lecture to you. Nothing like learning something new, right? So brown algae are Stromenopiles. This is a group of eukaryotes that we'll talk about a little bit, and two Archaeplasidae, which are the reds and greens. And the greens, of course, led to land plants. I was trying to think of a typical and charismatic New Zealand alga, and I guess it would have to be this thing. When you get old, as I am, you start getting grouchy, and this is probably about the most iconic alga of New Zealand that I can think of, and this is what I get grouchy about. It was the only seaweed on a monetary note, and you can see at the top we have a penguin and birds and a penguin and some plants and some Dervilia right here, and they redid it, and they kept the bird, they kept the other one plant, they kept the other plant, and they got rid of the alga. This is kind of the position that phycologists have to be in. They obviously didn't consult me. This is just some work looking at diversity. So this is an alga that's very common in New Zealand called Lawsonia. It was thought to be one species and all over the country. It's an alga that makes alginate. It's got a possibility of being grown for alginate, which is a cell wall product that can be used commercially. This is what it looks like. It turns out that if you look at it genetically, there are 4 species in New Zealand that have very distinct distributions. So there's one that goes up to East Cape. There's one that's found in Kaikoura. Genetic diversity, multiple species. If you want to harvest these, if you want to exploit them, you have to take this into consideration. Then there's another brown alga, that Carpophyllum. They don't really have common names. This is called rack. There's so many things called rack, it doesn't make any sense. So this is what it looks like. It also got floating devices. And you can look at the genetic diversity, so just the colors. When there's more color, different colors, they're genetically different. And you can see that in the south and the east coast, there's less genetic diversity. And there's reasons for this. It has to do with climate change, and there's changes in seawater temperatures, and these things have been moving down as the water has been cooling. As the water has been warming, but different genetic diversity. This is a red alga. These are the sort of studies I do very often. It's just trying to understand the genetic diversity of these organisms and trying to get some idea of what we have in New Zealand and how they live. So here's an alga. Just look at the colors. There's some purple species here. They all look the same, genetic types, and then there's a purple one. And in some places, there are multiple living together. And the question is, when organisms live together, what do? Do they segregate the environment? And it turns out in this organism, it does. So this is an alga that grows in the intertidal. It's called Apostrichia intricata. In one genotype, so the the orange genotype grows much lower than the other genotype. So genetic diversity is not just a measure of some random thing. It's a measure that reflects some ecological characteristic. Its production, its productivity, its reproduction, and also how it segregates in the environment. So that's one of the reasons that we try and study these things. I also had the opportunity to work with a colleague that couldn't come here. His car broke down on the way. He lives in Raumati now. And to work on a green alga. I don't usually work on green algae, that grows in Lake Baikal. But Lake Baikal is in Siberia. Here it is. It's the largest freshwater body of water in the ocean. It's the deepest, largest, oldest, and coldest. No, maybe not coldest. It's got all the epitaphs. Oldest, deepest, something. Very oligotrophic, very special body of water in East Germany, in Siberia, and it has a unique flora of green algae that have been studied by Soviet scientists, and that we wanted to look at also, and look at them genetically, because they've been described as different genera from what we studied before. And they're quite diverse. This doesn't seem like a lot of diversity, but for this group of organisms, it's a lot of diversity. So it's branched organisms, crustose organisms, ones that branch more, ones that branch less. This is as much diversity as you see within this whole order of these green algae. And it's a very unique environment. These are sponge green algal symbioses in this ocean. It's a very unique, very oligotrophic ocean of water. It has a landlocked seal thousands of miles from the ocean that got stuck there, all kinds of interesting organisms, but also these things. But when we looked at them, instead of being different genera, it turned out that they were all very closely genetically related to each other, and they were all nested within a group of organisms that looks very similar. They're all unbranched filaments. So this morphological diversity seems to have originated in Lake Baikal. And Christian and I applied for a Marsden, and we got some money to continue this work. So they look different. Are they different species? Or is it just genetic variation within a species? The resolution of the markers we used at the beginning were very low. And so Christian and a PhD student continued on this. and showed that these species, many of these morphological types are actually genetic species. And this is called sympatric speciation, which is very rare in biology, or at least rare to show in biology. Organisms that live in the same place that speciate, and certainly not known in algae. So here's just this, these are the individuals around these rows, and the color puts them, based on genetic data, into one species or another. So here you can see a morphology that all fits together, and here's a morphology that mostly fits together, and some don't quite match. The morphology doesn't quite match the genetics. So that means that there's some sort of reproductive compatibility between these individuals. This is rhizoclonium, these Baikal samples. And then, of course, there's this, right? There's the continued, and something that I spend quite a bit of time with some of my colleagues, especially at NIWA. There are things that look different, they're all the same, and there are, of course, things that are the same that are different. And so just to untangle that, again, if we don't know what they are, if we can't give them a name, then how can we understand their distribution? How can we understand the chemistry that they have? How can we understand how they'll grow and be impacted by the environment? These are all the same thing. I would never have guessed it, right? Genetically, they're identical, right? Again, this is an ongoing, we're very lucky in New Zealand for lucky, maybe is the wrong word. We have lots and lots of species that have not been described in New Zealand, and we still don't understand the diversity that exists in New Zealand. Okay. I want to kind of change a little bit and talk about chance, serendipity. And this is sort of backing up this idea that I had of the ability you have as an academic to just follow things that you're interested in. Having students, having small amounts of money paid for by given by the university to do research of whatever interests you. Right? And so it's called serendipity. So my first serendipity is, in one of my classes, we do a DNA extraction, and we sequence a seaweed, and I teach the students both lab techniques, but also how to understand diversity based on DNA sequences. And so we do a DNA extraction. And when we do a DNA extraction, you expect a heavy, high molecular weight band. These are the chromosomal big band of DNA. And all the students do that, and they all run the gel. They're all happy because they run agarose gels and put blue juice into agarose gels. But this one had a band. It actually had two bands. It had a band there. And this is from a red alga called Champion Nova Zelandia. And, it's like, there are 60 students, lots of labs. It's like, yeah. I looked at it and I said, that's interesting. I wonder what that is. 2 honor students later, we've kind of figured out what it was. For a long time, I thought it was a plasmid. So red algae are known to have plasmids. These are circular extrachromosomal DNA that exists in the organisms. This is a DNA extraction, a lab DNA extraction, that uses RNase. So I thought, okay, it can't be RNA. But so this is the same, a different gel. Here's a ladder. This is the DNA, the high molecular weight DNA. Here's that band that I was talking to. If you add a DNase, an enzyme that eats DNA, it actually removes that, but not this. That means it can't be DNA. After a long process, we learned out it was a double-stranded RNA virus. And it's actually, so we sequenced it, it's made up of 2 genes, and it's a totovirus, which are known in some red algae. But the most intriguing thing is when you run this virus in a DNA extraction, you have a band, and then you have a band you can see with your eye. It turns out that this totovirus makes up about 30% of the double-stranded nucleic acid in this organism. I mean, I don't know where that could possibly happen in the world. Right? It just is mind-boggling. What is it doing, right, to copy itself to 30% of the nucleic acid that exists in the organism? Right? How does it affect that organism? So this is individual plants, the percentage of double-stranded RNA within those individual plants. A large amount. I'm not sure I will continue with this. Those questions may be very difficult to answer, but just that they have it is intriguing, right? And it all led from just me paying attention to when students ran these gels extracting from random algae in New Zealand, right? A little bit of money to do that. A while ago, I had a friend who worked for a company, a chemical company, Degussa, which does all kinds of chemistry, including carbohydrate chemistry, and red algae. This is another red alga. Red algae are, even though it looks green, red algae have products, carrageenan in this case, which is used for industry, for all kinds of things, additives to food, grown in the tropics like crazy. This person had all these dried samples. I had some DNA markers that would tell variation within species. And he said, could I send these to you to sequence? I'm like, sure, it'd be fine. It wouldn't cost that much. So he sent me, 200 samples. Spent a bit of time sequencing them. Are these the same species? Right? They all look different. Here's these farms, gigantic farms. This is in Indonesia, but in Malaysia and the Philippines, and now all over the world. It's been sequenced because you can put it into food. Right, here it is, carrageenan. If you have anything slimy, fast food, frozen dinners, they'll have carrageenan in them. Toothpaste will have carrageenan in it. It's just slime from a red alga. But it's a big business, and there's also the social benefits of farmers who are fishing to actually grow these things. And so I ended up sequencing these. Some of them were wild, some of them were cultivated. All the cultivated ones were practically identical, so they're identical with the markers I had. This is what this line up and down means. Here's another Eucuma denticulatum, also identical. It looks like in farms all over the world, including in the Americas, they're identically genetically. Which makes sense. You just break a piece off, you actually put it in your pocket, get on the plane, take it to some place and put it in the ocean and grow it again. Right? So there's no genetic variation. And this is starting to cause a problem. With no genetic variation in monoculture, you're starting to have a problem of diseases. Right? And we have to learn to increase the genetic diversity. Before we didn't know that there was no genetic diversity. We could have guessed it, but we didn't know it. Not only that, these things in farms are all identical. So this is in Africa, so this is Tanzania, this is Zanzibar and Pemba, 2 islands off of Tanzania. In farms, they're all the genotype that is used for farms. In outside farms, in coral reefs, in seagrass beds, the African genotypes are disappearing because these plants are breaking off from farms and spreading to outside the reef and outcompeting, just because of numbers, outcompeting the genotypes that you would like to keep if you want to produce plants that will have higher genetic variation to protect them against diseases, for example. So this was continued work with some people from Sweden after I'd written the first paper. And of course, this led to a bunch of work in Indonesia. I've taught 3 workshops in Indonesia. They really care about these things because they have carrageenan. Teach people how to do basic molecular biology, genotyping these samples to try and understand the genetic variation and what species they're growing. Mostly farmers don't even know. Right? They just grows well. So here's me in one of the workshops. I was going to go to one just before COVID hit. So I missed one out, and here's me talking to somebody who knows what the heck I'm saying, but here I am eating Indonesian food, which is very nice. Here's another kind of serendipitous story. So I've been working with some people in Asia, in Korea, on something I'll talk about at the end, which is a bit of genetic variation. This is me at a porphyra farm, a pyropia farm. So this is a nori farm, or kim as it's called in Korea, farm. They grow it in Korea. This is it. This is the stuff that gets wrapped around your sushi. It's a big business in Korea. It's more profitable than all the other aquaculture combined. Again, have problems with diseases. This is a picture around Wando from an island. All these stripes are those nets. Right? This is just a small part of these maps. Big business. There's always ideas of trying to bring something like this to New Zealand. We have lots of these related species to these ones that are cultivated in Korea, in New Zealand. This is just me and my colleagues. You have to go in winter. They don't grow in summer. And of course it has diseases. And one of the diseases is called red spot. And so this is the red spot disease. It kills them. They don't look nice. The price goes down. The plant falls apart. And so my question was, I was there for other reasons. I thought, well, I wonder if they have it in New Zealand. We have lots of these algae in New Zealand and related things. So this is actually red spot. This is what it causes. It just kills them. Turns out it's a stromatopyle. It's an oomycete. And I had a student look at it in New Zealand, collected from different plants. It exists. You can isolate it. You can grow it in dishes. So oomycetes. Oomycetes used to be called lower fungi. Never understood what that meant. They used to be called pseudo-fungi. I don't understand what that meant. They're not even either. Right? They're just stromatopyles, but they cause diseases. Same group of organisms causes cowrie dieback and Phytophthora disease in potatoes and things. And of course, it's the same. So in New Zealand, we have this genotype that's the same as the ones that are causing diseases in Korea. And of course, this is something we have to think about, right? If we want to have an industry of pyropia, and it turns out that this thing will infect other pyropia plants. If we want to have an industry, we have to take into consideration that the pathogen is already here. How is that going to affect things? What are we going to do about that? Okay. All fields of science have a bit of esoterica. Esoterica in novels is always interesting. We have esoterica in science, kind of how it progresses. We just talk to each other about things that are important to advance the field that may not be interesting to other people. And I'm going to talk a little bit about esoterica and how it led to an understanding of something that I think is more important. So, again, teaching you, red algae transfer nuclei between cells not from the same division. It's called secondary pit connection formation. And it produces this beautiful pattern where you have cells that have one nucleus and cells that have two. It's a normal process that red algae have. But also, after fertilization, they transfer nuclei. So this is an egg. This is the extension of the egg. And these are 3 sperm attached to the egg. They fertilize that egg, makes a diploid nucleus, and that diploid nucleus goes somewhere. It often goes to a nearby cell. It's got special names, like all reproduction. It has crazy names. It goes to another cell and then causes that cell that used to be haploid, that has this new nucleus, to act differently. Right? It grows into a zygote with multiple cells. It grows into a diploid that's parasitizing on the female. And this work has been going on for a very long time. You can always tell that is because it's written in German. That's how old it's been. But again, these nuclei get transferred. Very odd. And this is just some work I did with my Korean colleagues trying to understand how the auxiliary cell, the specialized cell, gets ready to accept that nucleus. So here we have a picture of an egg with a sperm attached. This is a reactive oxygen species. Very interesting. So these products, which are really poisons that are produced by chloroplasts and mitochondria, are actually now used as a signaling molecule. So here's the molecule. The sperm is fertilized. Once the sperm and the egg fuse, the body of the egg becomes labeled with these reactive oxygen species, and the cells that maintain that hold that egg also get labeled. And here you can see it. The egg gets fertilized, and these cells get labeled. Why are they doing that? Well, again, it's this thing. In this organism, the signal seems to go down these cells and activates a cell division to prepare a cell to accept that diploid nucleus. Esoterica. It gets weirder than that. These diploid cells can transfer that nucleus to another diploid cell that can grow into a multiple diploid, parasitizing the female, and be transferred to another cell. And they just pass on this nucleus, and they grow into a diploid on the female and just release these things. Very odd. It's a thing that red algae do and nobody else does. And of course, esoterica, you have to interpret these kind of things. I wouldn't try. I can almost understand it. Roberta is here. She's the only one that can actually do this work. But you can see just what I said. So here's a cell that has received a diploid nucleus through this cell. Here are these cells transferring these diploid nuclei to a special cell that then grows and continues growing and releases these diploid nuclei. So basically, from one fertilization, you can get millions of diploid zygotes released. It's a very unique characteristic of red algae. Why is that interesting? It's because this process has been kind of hijacked somehow by parasites. So there are red algae that are parasites on other red algae. And they've hijacked this ability to transfer nuclei to a cell that they're not related to and get it to act like one of them. This is just some pictures of those, some very nice ones. There's lots of them in New Zealand. It turns out that these parasites, so these are parasites in blue and the host in red, the parasites are more closely related to their host than the host is to other congenerics and conspecifics. These parasites came out of the host that they grow on. Right? Sometimes they're not so closely related, so they're in the same genus, but they're slightly more distantly related. They share a common ancestry further back. And what they do is just what it sounds like. They transfer a nucleus. So this is the parasite in red, transfers a nucleus into a host cell. And in this case, the host cell just grows and acts as a sink for nutrients. Here's some pictures of that. Here are the host nuclei, and here's the transferred parasite nucleus. It transfers a nucleus into a non-related cell and gets it to do something for itself. And of course that continues. There's some parasites that will transfer that nucleus, and that diploid cell starts, the nuclei of the parasite start growing. They continue growing. They infect other host cells. It produces a parasite that then becomes reproductive. These parasites don't really affect the host. There are about 120 independent evolutions of these parasites. They seem to have hijacked this machinery of passing nuclei around and controlling somebody who's not you, right? Using their nuclei to control things. How they came about, what the effects of all this is, are just interesting questions for a phycologist. Here's just more pictures of that. Here's a parasite fused to this host cell. This is the host nucleus, and these are the parasite nuclei in the cell. And it transforms them. So here's a parasite passing it on to a host. This host has chloroplasts, these yellow things, but they're starting to be degraded. Right? It's turning this host cell into a parasite cell that then grows differently. So what's the outcome of all that? Right? What happens to these organelles that get transferred into the nucleus? They got nuclei. What happens to the host nuclei? What happens to the host mitochondria and chloroplast? The same for the parasite. What gets transmitted to the next generation? Does everything get transmitted to the next generation? And of course, how did it all come about? Right? You've got this idea from this esoteric understanding of parasites, of red algae transferring nuclei between unrelated cells, but somehow it got co-opted into this process, and how it came about. Question to keep me alive, keep me thinking about it for a while. So New Zealand has lots of parasites, new discoveries all the time. There's lots of questions about what species we have in New Zealand, what variation we have in New Zealand, what evolutionary transitions we have between different species, what physiological capacities we have about this underlying genetic variation that we can't see from just morphology. And also, it's just important to be able to have the opportunity, not only mentally open and just observing things without thinking about the consequences. Yeah, I'm apparently told that I always do things without thinking about the consequences. My wife keeps telling me this. But also the freedom to pursue these things, right? And so universities do provide this ability, right? I'm here to teach students. I'm here to train other scientists. But I also have the ability to just follow my interests, right? To advance the field, without thinking about where it might go, right? Just because it's interesting and it's advancing my research field. This is a saying by Theodor Boveri. Boveri is a very famous biologist, German. You can never misquote people or take quotes out of context. The first half of this quote is actually very good. Boveri discovered that nuclei-controlled development. He also was the first to try and make the connection between chromosomal abnormality and cancer. He also was a very strong advocate of marine stations and spent every summer. Why would you spend it in Germany? We can go to Naples to Anton Dohr Marine Station. And so he had a very strong connection with the marine stations in Naples. Equally strong is the contrast between the marvelous freedom of research, which is the good part of that, and then the other part, never end on a good note, and you can't take it out of context, but research one finds interesting, and the sufficiently exercised task to press zoology, I could say phycology, into heads that do not want to know anything at all about it. I didn't want to take it out of context, so I'll give you the whole thing. This is a letter he wrote to Anton Doorn. And thank you very much. Oh, before I forget,
Wonderful. Thank you very much. Tēnā koutou katoa. My name is Ehsan Mesbahi, and I'm the Pro-Vice Chancellor of the Wellington Faculties of Science, Health, Engineering, Architecture and Design Innovation, also known as SHEDI. And it's my great pleasure to give the vote of thanks to Professor Joe Zuccarello for tonight's lecture. I have to go a little bit off the script, if I may, because Joe is a marine biologist and I'm a marine engineer. So I started working on algae because I wanted to kill them. Because algae and different type of phytoplankton get on board ships and go around the world. And they call them introduction of non-indigenous species, and they cause a lot of problems because sometimes they produce toxic algal blooms. So my job was to find the best way to eradicate and kill them. And I had a research group of about 12 PhD students, and one of them, during the process of finding how you can kill algae and phytoplankton and seaweeds, he found the ways that you can grow them. You know, sometimes when you want to kill something, you understand how they live. So he started calling himself Dr. Seaweed, and he started a company in UK, Dr. Seaweed. I didn't know we have Professor Seaweed here, so probably, I don't want to put anything into your mind, but maybe you can do something business-like and... No, what he does, he produces seaweeds for nutritional and medicinal purposes, and he puts them in little capsules and sells them quite expensively. But anyway, so tonight we had the pleasure of having a very small glimpse of your fantastic enthusiasm for biology, and in particular marine biology, and the field known in the very reputable field of research with algal diversity, and this is something which I personally totally enjoyed. All these alongside your strong commitment to teaching means you have made and continue to make invaluable contribution to the School of Biological Sciences and the wider university community. As Vice Chancellor mentioned, your contribution goes beyond your research. At Teranga Waka, one of our primary purposes is to provide excellent education to our students and to prepare them for an extraordinary life. And this is something, Joe, that you do quite fantastically and successfully. You are an enthusiastic teacher, and we could probably see it quite clearly in the way you presented tonight. And you're always improving your own skills, you are inspiring our students, and it's not only looking into teaching biological information, but I think you also introduce critical thinking, and this is something which you probably repeatedly mentioned, the freedom of what you get at university, and that critical thinking without freedom is not helpful. And so I can see that how it has helped you and your students to flourish at university. I have to note here that his wife, Patricia, I don't know where she is. She must be somewhere. Hi, Patricia. Patricia is one of our senior student advisors in the Faculty of Science. And I think Joe admits that Patricia does a lot more contribution to university than you do. And I have to agree with him. I have to behind every successful professor is a very strong partner, and I think I have to probably thank Joe more than I'm thanking you for making sure that you're doing a fantastic job and with her support. And the fantastic job that Patricia does for the faculty and the division. Your lecture tonight also highlighted the important role that universities in general play in society. Our environment provides the staff with the freedom to explore a wide range of questions in the unencumbered pursuit of knowledge, while one of our primary goals is teaching, the teaching that inspires and prepares the students. Best is led by researchers who can feed their world-class research into their classrooms, and I think this is what you do brilliantly. Joe, as a Pro-Vice Chancellor, I'm very proud to have you as one of my professors. You have been around for 17 years here, and I hope that we can have you another 17 years at university. By the way, I've been here only 17 months, so I think I have a long way to go. And for this wonderful, fantastic inaugural lecture. And without further ado, I would like to invite all of you to not only join me in thanking Joe again for his fantastic lecture, but also there are some refreshments outside, and then let us go together and have some fun. Thank you, Joe.